This presentation will focus on topics related to environmental toxicology. By the end of this presentation, your reading of the chapter and the other online work that I ask you to do, I hope you'll be able to define the term toxicology, understand different definitions and important terms used in the field, identify 5 factors that affect responses to a chemical, and discuss and describe the steps involved in risk assessment, as well as the methods used for human exposure assessment. What's important for you to understand is that the reality is all substances are poisons. It's really only the dose that makes the poison. This is true of everything from water to medicines to, of course, chemicals that we think have very few reasons to be in the body, like lead. Toxicology is defined as the study of the adverse effects of chemicals on living organisms. Toxicology has a long history, and the father of toxicology is known as Paracelsus. He was really the one that coined the term, the dose makes the poison. And he contributed to understanding the ways that chemicals can cause effects by examining what's known as dose response. A toxicologist is a scientist who has received extensive training in understanding the adverse effects of chemicals at many levels, cellular, molecular, and assessing the probability of their occurrence. Environmental toxicology focuses on exposures to chemicals in the environment that may present risks to humans, animals, and other biologic organisms. Birds, fish, microorganisms, algae. Here are some key terms to be able to define, describe, and distinguish between. Poison is defined as any agent capable of producing a harmful response in a biological system. Toxicity is the degree to which something is poisonous. This is related to a material's physical and chemical properties. If a chemical can permeate the skin, it may be more hazardous than a chemical that can't permeate the skin. Toxicants are toxic substances that are man-made or result from human, also known as anthropogenic, activity. And a toxin usually refers to a toxic substance made by living organisms, plants, animals, microorganisms. For example, there is an illness called botulism, which can be caused by a bacteria called Clostridium botulinum. This bacteria produces a toxin when it grows. The toxin is extremely poisonous. Very, very low amounts of it can cause severe illness. That's an example of a toxin. Dose refers to the amount of a substance administered at one time. And importantly, when we think about dose in toxicology, we can think about lots of different definitions of dose. Exposure dose may be the amount that was in the air that you breathe. Absorbed dose might be the amount that gets past any barriers your body has and gets into the body. Administered dose, total dose, external dose, internal dose, and biologically effective doses are all different types of doses that relate to the penetration of the chemical into the body and its action in the body. And I'd like for you to take a minute and identify 3 of these and find a definition for them. So you can then compare and contrast the different types of doses that we describe in toxicology. Lethal dose 50 is a term used to examine how toxic a chemical is. And the definition is the dosage, which is the milligram of the chemical per kilogram of body weight, causing death in 50% of exposed animals. This term is related to how we might do toxicity testing in the laboratory. But importantly, the other thing we can do with this LD50 is use it to compare chemicals and how toxic they are. So for humans, the LD50 for sugar is very high, probably 1,000 milligrams per kilogram of body weight or more. The LD50 for something like botulinum toxin is very low, and it's how we compare the lethality or the severity of the outcome that the chemical can cause. Key to understanding toxicology is this dose-response relationship, where we examine the characteristics of exposure to the chemical and the effects caused by it. And we look at how increasing amounts of chemical exposure cause increasing amounts of illness. This dose response is a very important term that we'll come back to time and time again. You can use graphs to describe these, how exposure to a chemical results in impact. In these graphs, you'll see there's two different types. One that describes the response of an individual to a chemical, so what are all the different outcomes of that chemical, and the other one is how a population of people exposed to the chemical might react. In this first one, you see a dose response curve for a chemical exposure on the x-axis and the clinical response on the y-axis. And you can see that as the increase happens in the concentration or amount that the person is delivered, the clinical response goes up. At first they may be giddy, then they fall asleep, then they go into a deep sleep, unconsciousness, labored breathing, and death. This could be an example of let's say an anesthesia that's given to a person. At first they start to get happy, then they go to sleep, they go into a deep sleep. That may be where you would be having a surgery. Eventually, too much anesthesia could cause death. So along a dose for a certain chemical, you might have a response in an individual that follows all of these. Increasing dose, increasing severity of response. However, when we think about a population, we also have to think that there's variability in the population. And in this graph, what we're looking at is, again, dose on the x-axis and percent response on the y-axis. And so what we see here is that if our one outcome we're measuring is mortality or death from exposure to the chemical, then we look for at what dose do 10% of those being studied die, do 50%, which is the LD50, die, or do 90% die? And this is something we might do in an experiment with animals. This isn't data that we would ever gather from humans, but the information allows us to balance and understand and compare toxicity of chemicals. Important to understand, at certain types of chemical exposures, you may be exposed to some of the chemical and have no response at all. And that is called a threshold. Right up until that threshold where the lowest dose at which a particular response may occur, you could have exposure to that chemical and have no effects at all. There are important things to consider about chemicals and what affects the toxicity of them. One of the main ones is how they get into the body. Another one is how much was received, for how long, and whether or not that chemical interacts with other chemicals. In pharmacology, we know that certain chemicals and medicines can't be taken together, but this can also happen with environmental exposures as well. And lastly, every individual has different sensitivities. This could be associated with allergies, although less likely in toxicology to think about different allergies, but you can understand perhaps how some individuals have allergies or are sensitive to certain foods and others aren't. But individual sensitivity is what we see in that graph, looking at why 10% of a population exposed to a dose would die and others wouldn't. When we think about the most frequent ways that we are exposed to chemicals in the environment and they get into our body, we can think about ingestion through the intestinal tract, gastrointestinal tract, inhalation through the respiratory system, and dermal contact through the skin. Each one of these sites has barriers to reduce the amount of chemicals that get into the body. For example, in the respiratory system, in the nostrils, you have hair that stop particles from going into the throat and getting inhaled into the lungs. You also have things that would reduce particles from going deep into the lungs in the bronchi. In the gastrointestinal system, you have ways that the body will reduce chemical exposures by removing chemicals, and from the skin it acts as a good barrier for chemicals not to get into the body unless that chemical can pass through the skin. Routes of exposure include ingestion, where you're consuming contaminated food or drink, injections into the bloodstream, which would be most notable in a medical setting, contact with the surface of the skin. You may work in a place where you're having things touch the skin, or inhalation through respiration. When we think about toxic exposures, we can think about things that happen in an acute, subacute, subchronic, or chronic time frame. Acute usually is a single exposure for a short amount of time. It could be lethal if the dose was high enough. Subacute may be exposure for 1 month or less. Typically, the doses that would happen at a subacute level would be low enough not to cause severe illness right away. Or subchronic, which is exposure for 1 to 3 months. Again, if an exposure is causing a harmful outcome at this level, it's likely that it would be somewhat low because you wouldn't be able to tolerate it for that long. And chronic is exposure for more than 3 months. Typically, chronic exposure to chemicals occurs at very low levels where people are not feeling symptoms as a result of the exposure. Adverse effects of chemicals can happen at 3 different levels in the body. Local effects, damage at the site where the chemical first comes into contact. They can be much more distributed, where it's systemic effects throughout the body. Or target organ effects, where some chemicals can confine their effects to specific organs. One good example of this is lead. Lead is a neurotoxicant, which means it goes into the brain and causes more harmful effects there than in other parts of the body, although it can be harmful in other parts of the body as well. As we've discussed during environmental epidemiology, some exposures result in a very long time before you start to see effects. And this time period between initial exposure and a measurable response is called latency. It can range from a few seconds to several decades, especially for agents that cause cancer or are carcinogenic. And this is important because we often try to link exposures to chemicals to health outcomes that have happened decades later, and it's very hard to do. As I mentioned before, a chemical that causes cancer or unregulated proliferation of cells in the body is considered to be a carcinogen. We have lots of examples of these in the environment, and they are regulated with the most conservative or strictest measures in our federal regulations. How do we understand and learn about toxicity in toxicology? We can have volunteers, often who have had normal or accidental exposures, maybe give blood samples, urine samples, often during observational studies. We occasionally can pay people to be exposed to chemicals if the response is acute and reversible. Animals are often used for experiments, and more increasingly, we're using cells derived from humans, animals, or plants to do experiments. And there's also modern toxicology, which uses computational models to understand the potential for chemicals to interact with cells in the body. A key source of information from toxicology goes on to inform risk assessment. Risk assessment is a tool that we use in the government of the United States, like the Environmental Protection Agency, to provide quantitative or sometimes qualitative estimates of the likelihood of adverse effects that will result from exposure to a specific health hazard. We can do this for chemicals, we can do it for microorganisms, and it's an approach that allows us to estimate and ultimately protect against the potential for harmful outcomes. There are 4 main steps in risk assessment. The first is hazard identification. Identify the hazard, understand whether or not it needs to be evaluated and what the potential risks are. Then a dose response assessment, where we look at how increasing exposure to the hazard increases outcomes or risk response. Evaluate in exposure assessment how much of that chemical or microorganism is in the environment, what's the likelihood of exposure, the length of time people will be exposed, the estimated quantities that they'll be exposed to, and then characterizing that information by putting it all together to understand what is the actual risk of an outcome from the exposure. As I just described, hazard identification defines the hazard, examines the evidence associated if we're thinking about a toxic agent, and it produces an estimate of the strength of that evidence. Do we have epidemiologic studies in humans? Do we only have studies in animals? Do we just have studies in cells? Dose response measures the relationship between the amount of the exposure and the occurrence of unwanted health effects. Exposure assessment identifies the populations exposed, the magnitude, frequency, and duration of such exposures. It answers who or what is exposed, how does exposure occur, how much occurs, how often and for how long. We look to understanding concentrations of the chemicals in the environment, assessing exposure among populations. We monitor this. There's many chemicals in the environment we do measure and monitor to get an understanding of our risk. And then we can also just use computer modeling to estimate exposures. Risk characterization brings together all of this information to understand what might be expected at what level at a given amount of time. And lastly, we take steps to reduce exposures, to reduce risk through risk management. This helps us set standards, requirements for testing, and allows us to make guidelines and sometimes regulations about what's allowable and what's acceptable for the human population exposure to this chemical of concern.